video, I'm going to be talking about Chapter 15, Protein Movement Inside the Cell. So there are basically three ways that transport can happen throughout the cell. So one is through nuclear pores, another way is through membranes, so it could be chloroplasts, mitochondria, into the peroxisome, or from the cytosol into the ER. So those are a couple of ways that it can transport, especially through the membranes. And another way we have is called vesicular transport. So this happens through vesicles that can come from the outside to the inside of the cell. It could go from the Golgi apparatus to the endosome to the ER, and it's just a transport through vesicles. That's about it. So I'm specifically going to be talking about transport through nuclear pores in this video. I've drawn an animated version of how the protein comes inside the nucleus, just to save us time. So we have us here a protein, and it has this sorting signal, or also known as a nuclear localization signal. And if a protein has a localization signal, that basically helps it guide. That basically helps the protein guide itself into an organelle. If it has it, then it's able to go inside the nucleus or the ER or any other organelle. But if it doesn't have a localization signal, then it remains a cytosolic protein. So this one has a specific signal called a nuclear localization signal that will help it bring it inside to the nucleus, and it uses an import receptor protein, a nuclear import receptor protein that is again specific to the nucleus. Nuclear import receptor. And this protein will basically bind to the protein. I'll place it here. It'll bind to the nuclear import receptor, and this whole complex together will get stuck in these systolic fibrils that will basically help guide this whole complex into the nucleus and inside. So now that this whole complex is inside, we need to get rid of this protein, because we want to release it inside the nucleus, but it's bound, right? It's attached. So we have this other molecule called RAN-GTP that will basically bind to our import receptor, and it'll cause a conformational change that will cause this nuclear import receptor protein to be released into the nucleus. That will cause the protein to be released into the nucleus. So now we have our RAN-GTP and our import receptor bound. So RAN-GTP is going to allow it to leave the nucleus and into the cytosol. So now that it's outside, we need our import receptor protein to be able to go out into the cytosol, find more proteins, and bring it back inside the nucleus. And for that to happen, we need RAN-GTP to be, it needs to come off the import receptor. Because right now, the import receptor is not able to bind to any other proteins because RAN-GTP is bound. So we have another enzyme called RAN-GAP, which is an activating factor. Sorry for the messy handwriting, but it's called an activating factor, which will basically cleave off this GTP and make it into GDP. Actually, So now that that's GDP, RAN-GDP no longer has any affinity with the import receptor. So it's going to come off. It'll come off, leaving the import receptor to go and bind to other proteins and bring them in as well. So now that we have RAN GDP off, we need to have a higher concentration of RAN GDP inside of our nucleus. So RAN GDP will come into the nucleus, and there's another enzyme called RAN-GEF, which is an exchange factor that will exchange the GDP with GTP. So that's a T. With GTP. So this whole cycle basically is run by GTP hydrolysis, so that the import receptor is able to come in and go out of the cell through this whole process that is run by GTP hydrolysis. So I hope you guys learned something from this video. It was just a quick recap on the previous chapters. Thank you.